Okay, now we have done for the eukaryotic cells, organelles and structure and blah 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 for the previous topics. So now we look at these topics will be prokaryotic cell. We're going to look at what are the main differences between prokaryotic cells and eukaryotic cells. Okay, so what are the learning outcome that we are going to talk about here is, first, we're going to look at or outline the key structures. Okay, we want to look at the key structural features of a prokaryotic cells. Now, be careful here, because when you talk about the word prokaryotic cell in this topic, we are referring to bacterium only. Are you clear? We are referring to bacterium only, not other kinds of the prokaryotic cells. Later, I will show you guys what are the different kinds of prokaryotic cells and how they look different. Okay, so always remember in your topics, when talk about in AS topic, okay, when talk about prokaryotic cells, we refer to bacterial only. Okay, so basically they are unicellular, 1 to 5 micrometer in diameter, roughly. Generally, it can be slightly smaller, but generally they won't be larger than 5 micrometer. So they have cell wall, but the cell wall is the peptidoglycan cell wall. They do have the DNA, but it's circular DNA. They do have the ribosome, but they are 70S ribosome only. And the absence of the organelles surrounding by the double membrane means that they don't have mitochondria, they don't have chloroplast. Okay, and we are going to compare the structure of prokaryotic cell as found in the typical bacterium. Typical, guys, typical not means a special kind. Okay, with the structure of the typical eukaryotic cell in plants and animal. Okay, when we use the term prokaryotes or prokaryotic cells, generally means that they do not have nucleus. Are you clear? So the words pro means before. Karyons means nucleus. Before they have nucleus means that they are prokaryotic cells, means that they don't have the nucleus. Generally, they lack of membrane-bound organelle. For example, they don't have nucleus, they don't have mitochondria, they don't have chloroplast, okay, and even endoplasmic reticulum, you won't be able to see inside, okay, prokaryotic cells. And in general, when talk about prokaryotes, prokaryote can divide into two domains. Two domains mean two groups. One is called Archaean, and another one is called bacterial. And our focus is at bacterial only for today's topics, not Archaean, because Archaeans, the structural features actually a little bit difference if you compare between the Archaeans and bacterial. So don't want to confuse you guys. We're going to talk about bacterial only for today. So this symbol actually means for your information at this moment. But Archaean, we're going to learn them in detail when we go for the classifications of the living organism. Okay. Now, what's Archaean? Okay, so general, again, for information only. Archaeans generally, in the past, we group them as same as a bacterial under a group called Archaeabacterial. So Archaean manifests numerous differences in their biochemistry compared to the bacterial. Now we separate them. Initially, we put them together because there are no nucleus, so it means we put them together. But after the advancements of the technology, we realized that Archaea actually, they look so different from the bacterial. We decided to bring them into or put them into a new group, which we call as Archaea. Okay, Archaea were initially viewed as extremophile. What mean extremophile? Extremophile means they live in very, very harsh environment. For example, hot spring, where the surrounding temperature is so high, but they still can survive. If you try to imagine, I put you guys in a hot spring, maybe after 5 minutes you feel so hot and you like burn already, you come out, right? But these these prokaryotic cells, they survive in the hot spring. Or the salt lake means that with a very high concentration of the salts at the surrounding. But nowadays, we also found out that they live in a broad range of habitats. You can find them in the soil, oceans, marshlands, and as well as humans' colons. This is something special. The bacteria that survive inside the colons or large intestine is called as a methanophile. Methanophile is the one that actually produce the gas called methane gas. Okay, produce the methane gas, so allow us to fart, right? When we get, well, how come in the large intestine we have the gas come up? So this gas actually is a methane gas, right? Due to what? Due to this archaea that's right that survive inside our colons. Okay. So no clear example of the archaea pathogens or parasite as so far, but they often are mutualist or commensal, okay, in our body. Bacteria, very wide diversity in terms of shape. Okay, so some bacteria, they are spherical, known as cocci. Some of them rod-shaped, we term them as a bacilli. Okay, a small number of species even have a tetrahedral or cuboidal shape. Some of them, they exist as single cell, but some of them, they might pair up, but they're still unicellular. Even they pair up, they live individual. That means they live the independence. Okay, so for example, we have the diploids, means in the pair, they may form the chain. So we have the strepto. Strepto, the word strepto means the chain. Or staphylococcus. Staphylo means a bunch of grape or form a cluster. Okay, so what's this mean? If you look at diploid, means like this. Chain, it form like this. Okay, cluster, it form like this, like grape. Okay, so the words here, strepto, staphylo, basically means that's the descriptions. Okay, all for your information only. So bacteria can form complex association with other organisms. So we call it symbiotic associations. So we have parasitism, mutualism, and commensalism. So what's the difference between these three? In a study with the commensalism, commensalism basically means that one party gained the benefit, another party actually no loss. For example, I give you guys an assignment, 10 questions. So student A finished 10 questions, student B copied from the student A. So student A didn't lose anything, but student B gained something. This is mean commensalism. Are you clear? Student A do 10 questions, student B copy from student A. So student B gain something, student A no loss. Mutualism means that both gain benefits. For example, student A, same question, same thing, 10 questions. Student A do 5 questions. Student B do 5 questions, then exchange and copy. So both gain benefit. Okay? Talk about mutualism, both gain benefit. Parasitism means that one party gains, another party lose. So what does this mean? So student A completes the assignment, 10 questions. Student B take the question, take the assignment, the assignment, remove the name and put student B name. So student A lost the assignment, student B gained the assignment. So this is mean parasitism, or parasites generally. Okay. So the genome in the prokaryotes, this is very important. They have the DNA, but their DNA is a circular double-stranded DNA highlight. Okay. So generally, if I draw the structure later, okay. So first, this is the bacteria. Okay, what we describe here is the bacteria, yeah. Okay, we are talking about bacteria. Okay, so they have this circular double-stranded DNA in the main one. This is not a nucleus. You try to imagine in your room, you place the rubber bands. Okay, so this is what I mean by the DNA. Okay, so prokaryotes, they lack of nuclear envelope. So you can see that no nuclear envelope. And in addition, many important genes of prokaryote actually store as a separate circular DNA known as plasmids. So it means that they have the main, can you see that? This is the main circular DNA. Then they have the extra, some small DNA. So this one is termed as plasmid. Okay. And prokaryote, they lack of mitochondria and chloroplast. But how they carry out phosphorylations and this respiration? This is basically aerobic respiration. And how they carry out photosynthesis? They can take place across the prokaryotic cell membrane. Okay. So do they have cell wall? Yes, they do have cell wall. So for you guys, B only that we need to know is a peptidoglycan cell wall. Okay, highlight the term peptidoglycan. So when you label it, okay, later to detail, so means they do have cell wall. So I draw a cell wall for them now. So this is the peptidoglycan cell wall. for bacteria. And they do have the membrane. So this membrane sometimes will fold. Okay, I use different color. Let's say red color to draw the membrane. So they have membrane. Sometimes this membrane actually will fold like something like this. Can you see that? So the folding of the membrane is important for the enzymatic reaction. Okay, or certain reaction, for example, photosynthesis. Okay, they can carry out the respirations, aerobic respirations, or even nitrogen fixations that we learn in the nitrogen cycle. Okay, rhizobium, for example. Okay, so other informations, for example, you should know that plant, they use cellulose. Insects, okay, I mean, insect is not cell wall, right, basically, but fungi, they use a chitin. Okay, insect don't have cell wall. Don't to confuse you guys. Perhaps I can do something like this. Cancer, guys. Okay, cell wall found in plant, cellulose, or fungi. Okay, chitin. Don't want to put cancel this one. Here, chitin. Archaea, they do have cell wall, but they're different. The X layer, for example, S layer. So they have ribosome or not? Yes, they do have a ribosome, but their ribosome is highlight 70S ribosome or smaller ribosome. How they reproduce? They reproduce by binary fission. Okay, so do they have a genetic exchange or not? The answer is yes, they do have the genetic exchange. How? Through the horizontal gene transfer, okay, or we call it as a bacterial conjugation. So now I'm going to explain more about horizontal versus vertical gene transfer. So what's different? What are the differences between horizontal gene transfer and vertical gene transfer? What are the differences? Okay, so first, let's look at this. Bacteria, how they reproduce? Bacteria reproduce through the process called binary fission. So let's say this is a father. So they do not need to have a mother in this case, for example, or they have this is mother, they do not need to have father. Now, we do know that bacteria, they have the circular DNA, right? So what's this bacteria do? First thing, the bacteria are going to replicate their DNA. Okay, replicate the DNA. Okay, they replicate the DNA already. Okay, so what's the next thing they do? They carry out binary fission. Okay, they carry out binary fissions from one cells become two. Here, let me draw the cells, two bacterial cells. Okay, now this is why I just now copy this out. So therefore, I can put it in. Okay, so one copy and then one copy. So can you see that the parental cells now? Okay, can you see that the parental cells transfer the gene from the parents to the two daughter cells? Correct or not? Okay, so this process also termed as vertical gene transfer through binary fission, vertical down. Can you see that? Top down, right? From the father to the offspring, then offspring to the grandson, and so on. Okay? So this is what we call the horizontal gene transfer by the process of the binary fissions. Okay. Now, what this means by horizontal gene transfer? So for bacteria, they can actually exchange their genetic material. So how they exchange? So basically, yeah, the bacterial species A, black color. Okay, so this is their main genetic material. They do have a small circular plasmid here, circular DNA, extra one called plasmid. So this is bacterial species A. We can have the bacterial species B. different bacteria, blue color, for example, in this case. Now, in this case, nothing to do with the asexual reproduction at all. Okay, so they also have the plasmid. So what happened here is, okay, what happened here is they can establish a channel. Okay, they can establish the channel. Can you see that? And then they exchange. Okay, they can exchange the plasmid. So it means at the end, bacteria A got the blue plasmid at the end, and bacteria B got the black plasmid. Can I see that? So they exchange. So can I see the gene transfer, right? So this kind of gene transfer is known as horizontal gene transfer. And the process allowed this horizontal gene transfer is called bacterial conjugation. Are you clear? Bacterial conjugation. And we're going to learn this one in detail, how this process actually caused the antibiotic resistance to develop in the bacteria. Now they will say, oh, you can see that, oh, why so bacteria now become antibiotic resistant? They are not killed by a particular types of the antibiotics. Okay, so one question here, what is plasmid? Plasmid basically is the extra DNA or circular DNA material in the prokaryotic cell or in the bacteria. So generally they have the main one, but they do have the extra. The extra one is termed as the plasmid. They are circular, they are very, very small, and they can transfer between two cells or two bacterial cells by the process of bacterial conjugation. So allow this horizontal gene transfer to take place. Okay. Now we look at this structure of prokaryotic cells. As I said, that some structures of the prokaryotic cell always present, okay, in all bacterial cells, but sometimes certain structure only present in certain prokaryotic cell, not all. Okay, so let us look at the first one here. Structure that always present, okay, at the right hand side. So you can see the gray color. So this one is a cell wall. Okay, so when you label it, we label it as a peptidoglycan cell wall. It's not the plant kind of cellular cell wall. So we label it as a peptidoglycan cell wall. Okay, now second structure, after the cell wall, we do have the cell membranes, and the cell membrane may fold. Can you see that? May fold like this. Okay, so the second one is termed as the plasma membranes or cell surface membrane. Okay, now the third one here, actually I should point at the different parts, but accidentally point to ribosome. So it can be two answers now. It either can be the cytoplasm or ribosome. Okay, so in this case, cytoplasm. Perhaps you can actually lengthen the line a bit. Okay, if you put a ribosome, it's okay. Okay, so I should actually make it then better. Okay, so point to the cytoplasm. Now, what's the next one? So look at this. This is a circular, main circular DNA. So what's that? Circular DNA or bacterial circular DNA. And this one point to the ribosome. And to be more specific, you need to write 70S ribosome. So those are the structure that always present in prokaryotic cells. So what are the structure, additional structure here? Additional structure, for example, you can see that this one, some bacteria, they can move by actually have this flagellum. Okay, flagellum. Some bacteria may have additional protective capsule here. Okay, it's known as a capsule. Then you can see the infolding of the membranes. Okay, infolding of the membrane. So this one contains the enzymes, the infolding of plasma membrane, so that a certain reaction can take place. So what are the reactions? For example, respiration, photosynthesis, okay, can take place inside. Or some bacteria can carry out nitrogen fixation. Okay. Now, One thing that I want you guys to actually know here, the infolding of plasma membrane. Okay, let me draw out for you guys. Cell wall. Then you can see that sometimes they have the infolding of the membrane like this, right? And if this part is the one that carry out respiration, if only for respirations, then we term it as mesosome. The word mesosome is not in your syllabus, but come out before in the exam. Okay, come out before in the exam and ask you and write there mesosome, which is unfair for you guys definitely because you guys did. I mean, it's not in the syllabus, but just for information, mesosome means that the infolding of the membranes that contains the enzyme for the bacterial cells to carry out respiration. But photosynthesis, no special name. Nitrogen fixation, no special name. Mesosome only for respiration. Then as I say that they have this extracircular DNA, is known as plasmid, and they do have this structure known as pili for the bacterial conjugations to take place. Still remember just now I said that two bacteria, they connect to each other and exchange the plasmid. So how to connect? So they connect to each other by this pili. Okay, so any question guys regarding to this, the structure of the prokaryotic cells? Right? Next. So what are the differences between the prokaryotic cells and the eukaryotic cells? For the example, prokaryotic cell will be bacterial and archaea. Eukaryotic cell, we have the protoctis, fungi, plants, and animal. So at this moment, we need to know plants and animal only. Fungi, protoctis, we're going to learn this one in A2. Okay. In terms of average diameter, okay, our length, average diameter or length, basically it's length better, average length. So you can see that 1 to 5 micrometer for prokaryotic cells, sometimes 0.5, if some books use 0.5 to 5 micrometer. But for the eukaryotic cell can commonly up to 40, and it should be about 100,000 to 10,000 times the volume of the prokaryotic cells. Yeah. In terms of DNA, prokaryotic cell will be circular, life-free in the cytoplasm. Eukaryotic cell, not circular because linear DNA. Okay, and is contained in a nucleus. How about ribosome? Slightly smaller, 70S, compared to the eukaryotic cell, 80S. Now be careful. In this case, we are talking about ribosome, okay, at or in cytoplasm. Are you clear? Because we do know that eukaryotic cell also have 70S ribosome, which is inside the mitochondria and also chloroplast. Okay, endoplasmic reticulum, endoplasmic reticulum, no. For prokaryotic cell, eukaryotic cell, yes, got present. Cell organelles, very few cell organelles, and no separation of membrane-bound compartment unless formed by the infolding of the cell membrane. So inside this compartment, they can have an enzyme, or the enzyme can be located on the membrane. So it depends. Are you clear? So some protein and some enzyme may be located on the cell surface membrane, the infolding parts there. Okay. And we do know that compartmentalization takes place inside the eukaryotic cell. Cell wall, present in bacteria, highlight peptidoglycan cell wall. But cell wall in the eukaryotic cells, present, but sometimes, not all, only in plant and fungi. So plants, cellulose, you can write down. Okay, cellulose, not lace. Fungi, chitin. Okay, reproductions, binary fissions. Okay, now be careful. No, write down. No mitosis take place. Yeah, guys. It's straight away binary fissions. So DNA replication, binary fissions. Okay. But for eukaryotic cells, so involve the fusions of the male and female gametes. This is a sexual reproduction. Asexual, remember they have to go through mitosis. only followed by the binary fission for A. So asexual reproduction in eukaryotic cells must involve mitosis. Must involve mitosis for eukaryotic cells. Okay. Now, some extra things that I didn't put it in here. If let's say compared to the bacterial, okay, be careful when compared to bacterial. Bacterial does not form chromatid. No chromatid means that is only DNA. But for eukaryotic cell, we form chromatid. Basically means that our DNA will be together with histone protein. So this kind of DNA we call as a naked DNA. Okay, naked DNA means that DNA without histone protein. So this one we call as a DNA form chromatids. Okay, something that you can add on to the list. But only for bacteria, because Archaea, their DNA actually associate with histone protein for Archaea. But for bacteria, no, it's a naked DNA. Basically means that their DNA without the histone protein. So therefore, they cannot form chromatin at all. Okay. So we come to the last part for today. Or this lesson. We're going to look at this endosymbiosis theory. So what's this endosymbiosis theory? Let us look at how the transitions or how the evolution actually take place from prokaryotic cells to become eukaryotic cells. Okay, now let us look at this diagram. So first of all, you can see that the start of the living organism started with the prokaryotic cells, where you can see the DNA here. Okay, cytoplasm and also the plasma membrane. But towards the time, you can see that there is a growth of the cell become larger and larger, and you can see that the infolding of plasma membrane form the nuclear envelope. You see that? Form the nuclear envelope, okay, the infolding. And then this infolding continue to fold and form the endoplasmic reticulum, Golgi apparatus, and so on. Are you clear? Can you see that? Continue to inflow. Okay, the nuclear envelope, nuclear pore, and nucleus start to form in this case. But in the process of the growing or these are evolutions, phagocytosis take place. Phagocytosis take place, you can see that the cells actually engulf the bacterial. This bacterial actually is termed as the heterotrophic prokaryote. Now, what's heterotrophic? Heterotrophic basically means that they can't carry out photosynthesis. Are you clear? So this bacterial, they can carry out respirations. They do have the enzyme that carry out respiration. So this bacteria is engulfed by right. You still remember in the Golgi apparatus, we say that we have the lysosome, everything, right? Okay, to break down the bacterial. But the thing here is, our cells, ancestral cells, didn't do anything onto this. And in fact, this bacteria now cohabits with our cells and form our mitochondria. Can you see that? They enter into the cells. By right, our cells have to damage them, right? But the cells say, no, live together with me. So they cohabit. So endosymbiosis. Can you see that? Okay. So this is first time. First time we have the endosymbiosis. Okay. And then this group of cells split into two. One continue to form the eukaryotic animal cells. Another group carry out the second times of the endocytosis, phagocytosis take place. That means the bacterial, they can carry out photosynthesis. Can I see that? Okay. And this again, the cell didn't do any damage to this bacterial, and they live together and eventually form the plant cells. Animal cells, plant cells. Because mitochondria take place first. That's why animal cells and plant cell, both of us have mitochondria. But for plant cell, because after that they take in these chloroplasts, therefore, I mean the bacteria that form the chloroplast, therefore in animal cells you don't see any chloroplast, but plant cell, yes, because mitochondria take in first. So why scientists actually talk about this? So the endosymbiosis theory postulate that the mitochondria eukaryotic cells evolved from the Arabic bacterium living within the Archaeans, okay, Archaea host cells. And the chloroplast of the red algae, green algae, and the plant evolved from the endosymbiotic cyanobacteria live within the mitochondria containing eukaryotic host cells. So any significance or scientific evidence that actually supports the endosymbiosis theory or not? Let us look at this. So first of all, why scientists think that our mitochondria and also the plant chloroplast actually developed from the bacteria due to many evidences. First of all, if you look at mitochondria and chloroplast, weird because they are the two organelles that contain circular DNA. And this DNA is totally different from the DNA found in the cell nucleus, instead very similar to circular DNA of the bacteria. That's first evidence. Second evidence, if you look at the ribosome is 70S, same as the bacteria. And in terms of the size, it's quite similar because for the bacteria, it's roughly about 1 to 5. Mitochondria and chloroplast is about 1 to 10 micrometer, almost the same size as the prokaryotic cells or bacteria. Next, if we look at how the chloroplast actually multiplies, they multiply or reproduce through a process very similar to the bacteria, is by binary fission. Can you see that? Okay, and both mitochondria and chloroplast, they have double phospholipid bilayer. So why they have double? They appear to have arisen by the mitochondria and chloroplast entering eukaryotic cell while this endocytosis. When the mitochondria and chloroplast enter another cells by endocytosis, they are bound by the vesicle which form the outer layer of their phospholipid bilayer. Can you see that? Okay, so what's this mean? Now, let me draw it for you guys. Now, you look at this. When this is the eukaryotic cells, carry out phagocytosis, for example. This is eukaryotic cells. And what they engulf? They engulf the bacterial cell. So bacterial cells have their own cell membrane. Correct or not? Okay. So when the engulf completes, when this process completes, what will happen here is you can see that now this is the eukaryotic cell. They form a phagocytic vesicle, and then with the bacterial inside. Can you see that? So this layer will form later the outer membrane of the chloroplast and also the mitochondria. And then the degradations of the cell wall take place. Remove the cell wall. Oh, sorry. Remove. Wait, let me see what I can do. Yes, okay. Okay. Then remaining one is the cell membrane. So this one forms the inner membrane of the mitochondria as well as the chloroplast. But if the question asks any other evidence, yes, because any other evidence that do not support or does not support this endosymbiosis theory, yes, we do have, because where's the cell wall? But right, they have cell wall, right? But now no cell wall. Okay, but we can always explain in this case that cell wall may be degraded. Okay, so with this, I have done for the prokaryotic cells, and the checkpoint 1.19 is now open for you to go for the self checking. Okay.